Good morning. Today's topic is preparation of cDNA library. As we know that cDNA, it is prepared from mRNA. So, while preparing cDNA library, there are the three steps which are followed. First is preparation of cDNA from mRNA, because just now as I told you that cDNA is prepared from mRNA. So, this is the first step. Second is ligation of this DNA prepared from mRNA in a vector, and third is transformation of a host cell for the further propagation of the vector. So, first step, preparation of cDNA. First of all, we isolate the mRNA from a sample. Then this mRNA naturally it has poly A tail at its 3' end. So, this poly A tail will act as a known sequence for synthesizing the primer at the 3' end. So, we will take poly T as a primer for the synthesis of first strand of cDNA. Now, with the help of this primer and the enzyme reverse transcriptase, we will synthesize cDNA from mRNA's complementary DNA. So, we will get a hybrid of mRNA and cDNA. Now, in this duplex, we have to remove this mRNA and we need to keep only cDNA for further propagation. Then this mRNA is removed with the help of enzyme RNase H. This enzyme has the tendency of cutting mRNA in a RNA cDNA hybrid. So, this enzyme will delete this mRNA and we will get this single stranded DNA. Now, this single stranded DNA, its 3' end will turn on its own and will form a hairpin loop like a structure, and it will act as a primer for the synthesis of second strand. So, this 3' end having free OH group will be utilized by DNA polymerase enzyme for synthesizing the second strand of DNA. Now, using this end as a primer, this enzyme will synthesize the double stranded DNA molecule. Now, this loop structure, which is single stranded in nature, is needed to be cut down. So, for cutting this loop structure, we use single strand specific nuclease enzyme, which will make cut here and delete this portion. So, we will get a double stranded DNA molecule. So, till this step we have completed our first step of preparation of cDNA from mRNA, but in this process we are using single strand specific nuclease enzyme. So, many times there is a problem that the double stranded DNA which is constructed, it has lost its some of the portion due to the nuclease enzyme. So, this problem is overcome by a different method in which we used to construct, we used to find out, we used to select completely synthesized DNA. That is a separate process and it will be discussed later. So, after the preparation of this double stranded DNA, we will move to the second step, that is ligation of DNA in a vector. Ligation of DNA in vector. So, whatever is the size of this DNA molecule, according to its size, we will select a vector and we will ligate our double stranded DNA molecule into the vector. This is done so that the DNA which is constructed can be further replicated in the vector, so that it can be propagated further. Now, after the DNA is ligated in a vector, this vector is need some living cell, living system, so that it can further replicate and this gene can be propagated. So, in order to maintain the existence of a vector and to increase its copy number, we need to transfer the vector into a host cell. So, now we will move to the third step, that is transformation. In this step, generally the host cell taken is E. coli. It is not necessary that E. coli is taken, other cells can also be taken, but generally E. coli is taken. Now this vector which is constructed earlier, this is host cell, it is transferred into the host cell. It can be transferred by different methods like calcium chloride method or electroporation. There are many different methods which can be used for transformation. So now after transformation, the vector gets transferred into the host cell, and this is the DNA of interest which was transferred to the vector for further propagation. So in this way a cDNA library is constructed, and the initial mRNA molecule, it is changed into cDNA molecule because DNA is more stable in nature in comparison to RNA. So mRNA is changed to DNA, and then this DNA is transferred to a vector, it is ligated into a vector, and this vector after ligation of the DNA of interest is transferred to the host cell, and it is used to increase the copy number of the target DNA and to maintain the library of the target DNA. Now after, as we have read about the cDNA library, there are two types of libraries, genomic library and cDNA library. So what is the application of cDNA library? Why we are constructing a cDNA library? As we already have an option of having genomic library. So application of cDNA library is that if you want to express any kind of a eukaryotic gene in a prokaryotic system, expression of a eukaryotic gene in a prokaryote, it can be done by using cDNA library because next point is its property, because it has the property that the cDNA which is obtained from mRNA, it does not have introns. And suppose for example, talking about the application, suppose we want we in our general life we say that in markets insulin is available. So how it is available? The gene of insulin, it is ligated in a vector and it is transferred to bacterial cells and it is grown in fermenters. So how a eukaryotic gene is transferred to a prokaryotic system and it is expressing there also? It is because we are taking cDNA of insulin gene and we are transferring it to prokaryotic system, and a eukaryotic gene is expressing well in a prokaryotic system because this gene is taken from mRNA and it does not have introns, because a prokaryotic system does not have enzymes for splicing of introns. So the gene which we are taking is already free of introns and we can use that gene for expressing in a prokaryotic system. So this is the application, and in this application we are using the property of a cDNA, that it is free from introns. This property of cDNA is acting as an application in this system. Also there are many other properties like cDNA obtained is smaller in size in comparison to its respective genomic library, because it does not have introns, uh, and all about is. Thank you.